Bismillahirrahmanirrahim, November 24, 4-2. This paper is for 1 hour and it is for 40 marks. So you must know. As some of you have commented in my on the YouTube videos that I should specify the grade threshold. Now the grade threshold, of course, here for paper 4-2, you can see here getting a 29 out of 40 was an A grade, and getting a 23 would mean a B grade, and 18 would get you a C grade, and then 14 would get you a D grade, and then 11 would get you an E grade. So knowing that the grade threshold, you should always think of this when you are attempting the exam and see for yourself how you can make lesser and lesser careless mistakes and manage to get the grade that you want. Question number 1. Carbohydrates are found in different plant organs in the form of starch and sugar, such as glucose and maltose. A student decided to test two different plant organs, a potato and an apple, for the presence of starch and sugar. Suggest a suitable reagent for the test of starch and describe how it should be used. Details of possible results are not required. So what you do is you crush the sample. Why do you crush the sample? Now some of you come up with weird answers like increases the surface area or anything. Why would you crush a potato or why would you crush an apple? Because you see, if you crush it, the cell contents are released. The cell contains starch or the cell contains glucose. So when you crush it, you crush the wall and the membrane, and hopefully the cell contents will be released. So you crush the sample, number one, and number two, you add iodine solution. Iodine is not in a powder form, it's in a solution form. In fact, it's a potassium iodide solution. That was all. You didn't have to tell me that it turned from a brown to a blue-black color, because it says possible results are not required. But I'm sure many of you wrote that because you didn't even bother to read the question. Then it says the student carried out the test for starch on a piece of the potato and the apple. Figure 1.1 shows the appearance of the plant species before the test and after the test. So this is what they look like before the test, and then this is what they look like after the test. So you can see the potato is all black. And then it says, record in table 1.1 what you observe in figure 1.1 after the test and what you can conclude. So what was the observation for the apple? It was partially black. So it was partially black. It wasn't all black. This is partially black. And the other one was fully black or completely black. Like so, what is the conclusion? Less starch than potato. And starch present. So less starch than potato, or what could you have said? A bit of starch, some starch. Could have said any of those things. Less starch, you could have said. You could have said less starch than potato, bit of starch, some starch, small amount of starch. So anything of that. And this, of course, was your. You see, you must remember what is observation. Observation is what you can see. Conclusion is okay. What is your conclusion? Is there protein present, starch present, reducing sugar present? That is the conclusion. So this is what you've got to understand. For 4 marks, you have to give me 4 points for that. Then it says the B part of the question, the student then carried out a test for sugar on the potato and the apple using Benedict's solution and allow him following these instructions. Cut 1 centimeter into 1 centimeter cube of potato. So you cut a cube of potato, which is 1 centimeter by 1 centimeter. And on a white tile, using a cutting device to cut the tube into small pieces and place these in a large test tube. After 4 centimeter, add 4 centimeter cube of distilled water and use a stirring rod to gently crush the pieces and mix them with the water. Clean your tile, cutting device and stirring rod. Repeat this procedure with the apple. Add 8 centimeter of Benedict solution to both test tubes. The contents of both test tubes will be blue. Heat the two test tubes for 10 minutes at a temperature between 75 and 85 degrees Celsius. Describe how you would keep Describe how you would keep the test tubes at the required temperature for 10 minutes. Well, of course, how would you do it? The first thing is you say keep it in a water bath, or you can say thermostatically controlled water bath, and that would get you the 2 marks. And you use a thermometer to check the temperature. So if you said a thermostatically controlled water bath, well, then you got 2 marks for that. But if you just said water bath, then you would only get 1 mark for that. And then you could have said a thermometer to check the temperature. Thermometer word is underlined, so that means you couldn't have used anything else. Said use an apparatus to check temperature. No, we said use a thermometer to check temperature, or use a thermostat to control temperature. Then part two of the question says after 10 minutes the student observed the test tube. The color of the apple mixture was red and the color of the potato mixture was blue. So it says state what conclusions can be made. Now the typical mistakes that you will be writing will be you will not be answer you won't be answering the conclusion, but you'll probably write some sort of an observation or something like that. So apple shows that sugar is present. In fact, if I would say, I mean, if this was A levels, I would not accept that. I would say reducing sugar present, because there are two types of sugars, reducing and non-reducing sugars present. And in potato, there is no reducing sugar present. And both of these got you 1 mark. No reducing sugar present. So, I mean, of course, I'm being a little overzealous because I teach the A levels as well. So you could have said sugar present and no sugar present, no sugar present. So you would have got the mark. But I want you to remember to write reducing sugar because we're talking of reducing sugar. Now, coming to part three of the question, it says suggest why it was important to use a 1 cm, 1 cm, 1 cm cube for each plant organ because the results would be comparable. Number two, it says cut each cube into small pieces to increase the surface area, or you could have said to release the sugars or break open the cells. Of course, I prefer the ones of release the sugars and break open the cells. I somehow do not agree with the surface area part, but I can't argue with Cambridge. And then it says crush the small pieces and mix them with water so that you can release the sugars into a solution. So that the sugars come into a solution, or you can say form a sugar solution. So this is how we would word the answers, and this is a 3 mark question, and you have to be very careful not to lose a mark here. So the 1 centimeter, 1 centimeter was for results comparable, or the cut cubes into was increase surface area or release the sugars or break open the cells. And then the mixing them with water was to release the sugars into a solution or form a sugar solution. Now we come to the next part of the question. Explain why the tile, cutting device, and stirring rod were cleaned after using them on the potato. You prevent the transfer of the potato, or you can say prevent cross contamination. I mean, this English is very difficult. Some of you cannot sort of understand this English, but you must know this English. So basically, you don't want any cells of the potato to go and mix with the apple. So you want to prevent the transfer of potato, or maybe there's starch in the potato. You don't want that to go into the apple. You don't want any of the sugar to go into the apple, or you can say prevent cross contamination. Then coming to the C part of the question, describe how you test the apple for protein and state what you would observe if protein was present and not present. So the test is you cut or crush the apple and you add the biuret reagent. And if the protein present, it will turn purple, or you can say lilac or mauve or violet, many words for the word purple. And if protein is not present, then of course the solution remains blue because the reagent that you use is biuret reagent, and biuret reagent is blue in color. So it's very easy to remember. You use the word crush. Whenever they're saying a test, you're giving your solid, then you have to crush the sample and add biuret reagent. If the protein is present, it's purple. So very easy to remember. P for protein, P present, P purple. So it's three P's. And if protein is not present, then of course it remains blue. And I've written it in blue so that you all remember that. Now, coming to question number two, photosynthesis in green plants can be summarized by the equation carbon dioxide plus water in the presence of light and chlorophyll gives you carbohydrate and oxygen. The carbohydrate is of course glucose, which is of course not being mentioned here in this. And of course, another word which is not mentioned here is enzymes. I like to talk about that. We don't write enzymes, but in most of the biological reactions that we are studying, enzymes are involved. A student used the apparatus shown in figure 2.1 to investigate the rate of photosynthesis in an aquatic plant. Now you look at this very carefully, and there is sodium hydrogen carbonate solution because this is going to provide the carbon dioxide to this aquatic plant. You see, usually an aquatic plant would be in a pond, and in the pond there would be bacteria, there'd be fish, there'd be tadpoles, there'd be frogs, there'd be something in it, animals in it, which would be producing carbon dioxide for the photosynthesis to take place in the aquatic plant. But here we've just taken tap water, so we are adding a chemical to it which is going to be releasing the carbon dioxide needed by the photosynthetic plant. There is a blocked end of a 2 centimeter syringe, there's a bubble of oxygen, there's a cut end of the stem, there's a clamp, there's a glass jar, and there is lamp to provide the light, and there is the sodium hydrogen carbonate. When the student switched the lamp on, bubbles of oxygen formed and entered the syringe. The student recorded the volume of oxygen produced in 10 minutes. The syringe was then refilled with sodium hydrogen carbonate solution, and the oxygen collected for another 10 minutes. The student then decided to investigate the effect of different colors of light on the rate of photosynthesis. Leaving the same plant in the glass jar, a red transparent filter was wrapped around the glass jar so that the plant received red light. The volume of oxygen produced 10 minutes was measured. The measurement was then repeated. The red transparent filter was replaced by a green transparent filter and then by a blue transparent filter. For each filter, the volume of oxygen produced in 10 minutes was also measured twice. The measurements are shown in table 2.1. Now, what do we see in table 2.1? We see filter, no filter, measurement 1, 1.1, 1.3, and mean 1.2, red 0.7, 0.9, and 0.8, green 0.3, 0.3, and 0.3, blue 0.6, and nothing, nothing. The syringe at the end of 10 minutes of measurement 2 with a blue filter is shown in figure 2.2. Record the volume of oxygen produced in measurement 2 with a blue filter in table 2.1. You can see this is 0.4 because this is 1. This is 1, so you divide in 10 and then you see that 0.4. So we had to write that here, which is 0.4. And then of course you had to calculate the mean. 0.4 plus 0.4 plus 0.6. This is, 0.4 plus 0.6, so this is 1.0, and the mean divided by 2, so you will get 0.5. 1 divided by 2 will be 0.5. Basic maths, which of course none of us know. Then it said calculate the mean volume and record it in table 2.1. So we had to do two things in table 2.1, and we needed to write that. Then it says using the mean value, calculate the rate of photosynthesis per minute when no filter was used. Now, when no filter was used is this one, and the mean was 1.2, and we calculated that in 10 minutes. Rate is always per unit time. So 1.2 divided by 10, and this is 0.12. 0.12 would be the answer, but this 0.12 will not get you the rate. So it was 0.12 cm cube per minute. The whole thing you had to was get you 1 mark. There's only 1 mark. So cm cube per minute, or you had to wrote, even if you wrote per per minute, that was fine. So 0.12 cm cube per minute got you that 1 mark. Then it says B part 1, construct a bar chart on the grid to show the mean volume of gas calculated with no filter and with a different colored filter. So we have to plot the mean volume. So the rule that we follow is that the first column is the independent variable and that always goes on the x-axis. And the mean, this mean would go on the y-axis. That's the rule. First column is always the independent variable and the next one is the dependent variable which goes on the y-axis. Now we always have to label the x-axis and the y-axis and that gets you 1 mark. So axis fully labeled and bars labeled centrally. So you got a mark for that and the bars have been labeled here. You can see here green, red and centrally. Centrally means in the center of it. You write it here. And you can shade it if you want to. I would prefer you to shade it. They prefer this, so you can do a little bit of shading and put lines on it. So linear scale for the volume and value at origin, at least half of the grid used in both directions. So you had to use most of the grid in both directions. The mean values plotted correctly. So you have to plot these values which I've written here on the side. All bars ruled and of equal width and equal spacing and bars not touching. You see, it's a histogram in which you have the bar should be touching. Here I said equal width and equal spacing. I've even kept the space in it the same as the width of the bar. So please remember to be careful when you're drawing a bar chart because these are, as we say in Urdu, muft ke 4 marks hain. So it's easy to get these 4 marks, and anybody missing out these marks is a big time loser to not just draw this graph correctly. Then they've asked you in part 2, use the data given in the bar chart to state one conclusion that can be made from the results of this investigation. Of course, there are many conclusions, but basically all is green light, least photosynthesis, or you could have said different color cause different rates of photosynthesis, or you could have said normal light had the highest rate of photosynthesis when there was no filter. The normal light, when there was no filter, here you can see it here, there was the highest rate of photosynthesis. So any of these three, one of these three was, you'd get your 1 mark for that. So I'm just writing one of these, but you could have written any of them. Green light, less photosynthesis, or the lowest rate of photosynthesis, and different colors cause different rate of photosynthesis. Then coming to part C of the question, plan an investigation that you could do to determine the effect of light intensity on the rate of photosynthesis using the apparatus shown in figure 2.1. So same apparatus that we have here. And this of course would mean that you would not change the apparatus, you would use this apparatus which has been given in figure 2.1. Now looking at this apparatus, let's have a look at it. So we will say we'll start off because what are we changing is the light intensity. So at least three different light intensities. Now how could we change the light intensity? The light intensity could be changed by moving the lamp. Either you can say distance of the lamp moved, or you can say add a dimmer switch, or you could have said change the bulb. The bulb can be a higher wattage, a 60-watt bulb, a 100-watt bulb, a 120-watt bulb. Then, of course, you would use the same plant. You would use the same plant, and then you'd, of course, maintain the temperature. So, same temperature and the same concentration of sodium hydrogen carbonate. How much was the concentration? Was it a 1% solution or a 2%? I always say when you're talking about a liquid, then talk about the volume and the concentration. So if I say like 1% sodium hydrogen carbonate, okay, how much was the volume in it? Of course, here the volume has not been specified, but the volume you can specify in some other experiment. Then the syringe refilled for each light intensity. Then you allow the plant to equilibrate. And I've told you that you can write that in every planning experiment. Allow the plant to adjust to each light intensity, or you can say allow the plant to equilibrate. Equilibrate means to come into equilibrium with the environment. And for each light intensity, the next point would be for each light intensity, measure the volume of gas produced for the same length of time. Like here it was 10 minutes, so you would use the same thing. Whether you wanted to change it to 10 minutes or 15 minutes, that's up to you. So we can see the wordings of it now. Three different light intensities. Change. How would you change the light intensities? Three, change distance of the bulb, or you add a dimmer switch, or you change the bulb. And I said use a different wattage of the bulb. Then you use the same plant, then you use the same temperature, or you could have set the same concentration of sodium hydrogen carbonate solution. So that is only one mark if you measure temperature and that. Then syringe refilled for each light intensity, then allow the time for the plant to adjust to the light intensity, which means allow time to equilibrate. Please get your English right. Allow time for plant to equilibrate means to come into equilibrium with the environment. And then, of course, the last point is measure the volume of gas produced for the same length of time. So if you say you want to measure the volume for 10 minutes, or you say for 5 minutes, or for 15 minutes, well, please specify that. Now, coming to question number 3, figure 3.1 shows photographs of a male and female of an insect species. Both the male and the female insects are green in color. Now it says magnification is 3.5 times, and it says state one visible difference between the male and the female in figure 3.1. Well, there wasn't much of a difference. The one was the base of the back legs larger, or you could say thicker, or you could have said swollen in the male, this thingy here. So the back legs had something swollen in the male, and the female had a longer abdomen. You see, you can see this is a longer abdomen, while here in the male we have a wider or a longer abdomen in the female. So a wider or a longer abdomen in the female. So back leg swollen in the male, longer abdomen in the female, but you have to write only one of these to get the one mark. There are two different marking scheme points. Then it says in the space below, or make a large drawing of the male insect as it appears in figure 3.1. Now, most of you are going to scream at that and say, oh, miss, that's so difficult. But it's not difficult, it's only for 4 marks, and you can get all those 4 marks. You just have to be careful of what actually you see, you've got to draw that. Now, please remember when it says make a large drawing, so then it must occupy most of the space. I know my drawing is nothing very brilliant, but basically, what were the mark scheme points? The mark scheme points for 4 marks. How would you get the 4 marks? The 4 marks is male drawn, clear continuous lines, no shading, correct orientation. Then 75 millimeters in length. So that means the examiner would measure it. It shouldn't be a small diagram. It should be at least 75 millimeters in length from tip of the head to the abdomen. Then 6 leg drawn, double lines, at least first joint, and hind legs, femora, thighs swollen. Then 2 antennae attached to the head, and 2 wing cases clearly separate, shorter than the abdomen. So the wing cases that you had to draw, because the wings were folded onto the abdomen, so you had to show that. So 4 marks for this awful drawing. I'm sorry. I think my drawing is as bad as any of yours. And so you had to get these 4 marks. Please remember, clear continuous lines, no shading, and that is the most important thing. Then coming to part 3 of the question, C and D indicate the length of the female insect. D indicates the end of the abdomen. Draw a straight line to join C and D on the photograph. Measure the length of the line and record it. So you had to measure this here on C and D. Let's look at it here. So you had to measure this C and D. So this is from here to here. So you measure this and you show me the line on the diagram. That's about 31 millimeter. It could be different. You say have allowed anything between 29 and 31. So of course they give you a little freedom of a little point about 1 millimeter, or so anything between 29 and 31. Calculate the actual length of the nearest insect and record it to the nearest whole number. What does this mean, nearest whole number? I hope you all know that. So 31 divided by 3.5. Why am I dividing it by 3.5? Because the magnification was 3.5. So this thing, whatever it is, has been magnified 3.5 times. So if it is 31 millimeters, so it is 3.5 times larger. So that means you will divide it by 3.5. And then what comes out? The answer. So the answer, if you use your calculator, is 8.857. But then, of course, you can't say that. Nearest whole number would be 9. So this would be 9 millimeter. You can't write this. You would lose a mark. Please remember there are 3 marks. There's 1 mark for the nearest whole number. Then coming to part B, use the key below, identify and record the name of the insect. Wings longer than abdomen. No, they're not longer, they're shorter than the abdomen. So we go to 2 Then when you go down to 2, it says color brown, color green or gray. Now look here, it said in the question color was green, and this is 9 millimeter in length. So color is green, so when you go to 3, so you go to 3 then, and the length is somewhere between 7 and 11. So what is it? Is Edimera nobilis. So the name of the insect you had to write was Edimera nobilis. Name looks very regal, but then it's a beautiful insect. So you just have to write this here again. But if you had figured it out, if you didn't figure it out, well, then I'm sorry. You lose a mark. That completes this paper, and we have completed all the November 2024 papers. And thank you very much for subscribing, and thank you for watching.